Hi, everyone. Today we have Professor Wynn Legon, and he's at the University of Virginia. Wynn, maybe you could just tell us a little bit about yourself, what your trajectory was from graduate school through to your current position.
Yep, happy to. Thanks for having me, first of all. This should be fun. I did my graduate work at the University of Waterloo in the Department of Kinesiology, where we studied non-invasive neuromodulatory methods for mostly rehab and mostly stroke rehab. And then so I did a postdoc after my PhD at Sunnybrook Hospital in the neurology department, where we were looking at these non-invasive methods to kind of as adjuvant therapies for stroke rehab. And actually, which is interesting, and I didn't know at the time because I wasn't in the field of ultrasound, is that Kiervo Heinonen was just one floor above me, but I wasn't doing ultrasound at the time, and we weren't, ultrasound as a technology wasn't, I think it was nascent at that time. So we didn't have, even though he's he's the kind of the father of ultrasound neuromodulation, he was right above me and I had no idea. Anyways, so from that postdoc, I then went to Virginia Tech and studied with Jamie Tyler, where I got into ultrasound. And so this is where we started our human studies for focused ultrasound. And then since then, I was an assistant professor at the University of Minnesota, and now I'm at the University of Virginia.
Yeah, great. Thanks so much. We're pretty excited to hear you talk today about your in vivo applications. So thank you.
Great. All right. So today I will talk to you a bit about focused ultrasound for neuromodulation, and specifically its applications and use and methods for doing this in humans. So I have a background in non-invasive neuromodulation, particularly in humans. And I just thought I'd start out by talking about the other forms of neuromodulation and why the advantages and I suppose some disadvantages of ultrasound compared to these technologies. So I'm not sure if you're familiar with transcranial magnetic stimulation, TMS, or transcranial direct current stimulation, TDCS, but these are non-invasive methods to induce transient plastic changes in the human brain. And they've proven quite efficacious for general neuroscience research and also for clinical therapies. But they have limitations. And so what I'm showing here are some computer models of the electric field generated in the brain from these two technologies. And as you can see, they span several gyri. And so they lack spatial specificity. And so if you were interested in examining a specific cortical circuit or target, there's some potential that there's some overlap with other circuitry. So the point is that they're not spatially specific, at least to the size of a human gyrus. And then the other issue is they have a depth focality trade-off. So they're only really, the main part of their electric field is limited to the cortical surface, which you can see over here on the right. So anywhere is red, which is where the electric field is highest. And you can see that it doesn't extend very deep. And so it would be nice to have a neuromodulation technology that could go deep and is also spatially specific. And so I think that's one of the advantages of ultrasound. So I think you folks have already heard about or will hear about the use of ultrasound in different animal models. And I just thought I'd put up this slide just to show that prior to a lot of the human work that we've done, there was lots of animal work. And of course, this is also being done in parallel to our work. So ultrasound's been explored in mice and rats, rabbits, pigs, sheep, and in primate, looking at brain physiology and also different behaviors. So I won't go into those in too much depth, but I thought I'd put up this slide for a little context of where we are kind of in the chronology of ultrasound as a neuromodulation technology. So ultrasound for neuromodulation has been around since the 1920s, but it was only recently in 2008, 2010 that, at least in my opinion, that Jamie Tyler kind of reinvigorated the field with a couple findings, and specifically his finding in 2010 in this in vivo mouse model to show that ultrasound directed at the primary motor cortex can induce a peripheral motor response. And so you can see here in the blue bars, these are all the publications of the small animals that is continually growing. And you can see in the other colors represent the other animal models, large animal, primate, and human. And so this field is really starting to grow now. We're starting to learn a lot more in these different animal models. This is an unofficial count that I've done, so don't hold me to it, but I think it's representative of the current state of the field. So it's new. It's about a decade old, I would argue, and for currently, and so we've learned a lot, and there's still a lot to do. But I think the use of ultrasound in humans is ready and ripe.
When?
Yeah.
Yeah, just go back the slide. When did you take this data? Because it's really striking that the small animal research went down in 2020. I'm just wondering if you did that early in the year, or because it's October.
I did this.
Right? Yeah.
I did this the other evening while I was watching the football game.
And yeah, what do you attribute the fact that the small animal research has gone down so dramatically?
I think this is a COVID response. So that might be a little dramatic for the COVID response, but so I track when the papers are published. You can track these things on Google, and you get these updates through either PubMed or Google Scholar. And yeah, to date, I've only had five updates. So maybe that's not perfectly representative, but yeah, I hadn't thought about that. I just assumed that was a COVID, but it's not. Yeah, something hasn't dramatically happened that it doesn't work or people aren't studying anymore.
Okay.
Let's take the data out to 2019 and just we'll cut off the 2020 data. Okay. So the first study in humans was done by Stuart Hameroff and colleagues in Arizona, and so this was an interesting study where they used a clinical scanner, and they did this in some pain patients, and so they were interested to see what the effect was if they put a clinical scanner the side of the head and delivered ultrasound through the temporal window to target a portion of the temporal cortex. Did this have any effect on subjective report of mood or pain? And so this is a much different kind of application than what we do and what is generally currently done, but I'll leave it at that. And so what we do is we use single element transducers, which I know you're very familiar with from previous lectures. And in this case, in 2014, we directed ultrasound of the primary somatosensory cortex in humans to look for an effect on physiological measures as well as behavior. And we'll get into why we chose a somatosensory cortex as our first target. And really, it was for practical reasons. So I have a strong background in studying the somatosensory cortex. And one of the nice things about the somatosensory system is you can use what are called somatosensory evoked potentials to measure the response in the brain. And the advantage of these is that they have been source localized to particular areas of the brain, and you can record them non-invasively. So you can use surface EEG, scalp EEG to record these things. And so what I'm showing over here on the left is this is the dorsal column medial lemniscal system. So down here would be a periphery, and let's pretend what we do is we stimulate the nerve at the wrist. This is the median nerve, and so if you deliver a brief electric pulse to the wrist, it travels through this chain, a few different synapses in the medulla and the thalamus, and then it finally kind of terminates in the cortex at a particular area in the cortex. And we know this from ablation studies that so in primate, and this has also been done in humans, where you ablate a part of the cortex and one of these potentials will disappear, and this is how you would localize these things. So it's a nice methodology because we know where some of these potentials are generated. And so our hypothesis was, well, let's direct ultrasound where we know some of these potentials are generated and see if we can manipulate the magnitude of these things. And so this is not a new method, in the sense that lots of people have done different things to look at how the amplitude of these evoked potentials go up or down. And so what I'm showing here is this is I suppose a typical somatosensory evoked potential. And so these are all measured in microvolts. And you get these positive and negative inflections in the evoked potential. And they're named based on their polarity. So you have a P, and then they're based and also named on their latency. And this is the latency from the time of the stim at the wrist. So if I stim you at the wrist, it takes 14 milliseconds to get to the thalamus, which is here the P14, and then it would take roughly 20 milliseconds to get to the cortex. And then the assumption is they have these later inflections that also have these positive and negative polarities that are named, and I think I have this on the next slide. And so it's assumed that there's a serial processing chain in the somatosensory cortex that these potentials represent. It gets a little nebulous and a little murky as you go later down the chain, and I'll show you why that is. So our basic strategy then was to record surface EEG from 4 electrodes placed around the transducer over the somatosensory cortex. And we measured EEG continuously, just shown down here in the gray bar. And it looks like I've duplicated this here in the blue bar. And then you would deliver ultrasound time lock to that stimulus at the wrist. And in this case, we used a 500 millisecond ultrasound stimulus, which is represented here by this cyan bar. And you'll have to excuse me, I use different acronyms throughout this talk likely. So in this case, TFUS means transcranial focused ultrasound. I don't think the field's really come to consensus on what acronym to use, unfortunately, but they're all, I think, basically mean the same thing. So this is kind of a little brief crash course on the somatosensory cortex. So what I was saying is that these somatosensory potentials are generated in these different areas of the cortex. And so these numbers represent Brodmann areas. And so the CS is the central sulcus. And then so anterior to that is Brodmann area 4, which is the primary motor cortex. And then all these areas here, 3A, 3B, 1, 2, and 5 are areas of the somatosensory cortex. And so we know from these lesion studies that certain potentials, the N20 and the P27, are generated in area 3A and 3B. So let's direct ultrasound to those areas, and we should preferentially affect those potentials and not other potentials that aren't generated there. And so the issue as you go farther down the serial processing chain is you get more inputs. So in these monkey hands down here on the right, so you just get information from a single fingertip would go to say area 3A or 3B. But then to the later processing areas, you might get the whole hand or the limb. And so it's much more difficult to localize the later potentials. So we focused on the early potentials. Oh, here you go. So here I've just overlaid these potentials that I'm interested in. So the P14 is the thalamus, shown here, and then the N20, so it's negative, takes 20 milliseconds to get to the cortex, is the N20. Now there's a P27, which is 3B, and then a P50 or an N30, which is generated in the crown here of the postcentral gyrus. And then after that, it gets a little sketchy. So we wanted to concentrate on these potentials that we're quite confident where we know where they're generated. And so how a somatosensory evoked potential works is you deliver several stimuli to the nerve, and then you average that response in the head together to get this EP under the assumption that any random information will get averaged out, and then the evoked potential will then appear. And so what I'm showing here in these raster plots is these are the number of trials here on the y-axis from 0 to 100. And then on the x-axis is time. And then I've just color-coded each trial's effect. And then these two traces here are the averages. And so these would be evoked potentials. And then over here on the right is kind of just a top-down color-coded diagram of the spatial extent of different potentials at different latencies. So we have latencies as we go down here in the rows. And you can see that I guess one of the things I failed to mention that is if I stimulate your right wrist, this all happens on the left side of the head and vice versa. So I put this slide in here because this was a study we did in 2012, and also back here is studied in 2012. So instead of stimulating the median nerve of the wrist, actually what we did is we used ultrasound itself to generate these potentials. And so in this case, if you deliver ultrasound to the fingertip, you do get this brain response here. And actually these potentials here are evoked potentials, but from an ultrasound stimulus and not from an electrical stimulus. And so we could, if we change the parameters, we could change the evoked response in the head. So PUNS means peripheral ultrasound neurostimulation, and the M was this felt like a mechanical stimulation, whereas the T felt like a thermal stimulation. So you can change the parameters of ultrasound to increase temperature. And so that's what we were able to do with ultrasound, is get different waveforms based on the subjective feeling, and also the receptors that you stimulate with ultrasound. And so we did this in the MRI scanner as well. And I put this up here just to show that we do get responses in this primary somatosensory area, and then also responses in these different kind of hierarchical somatosensory areas in the brain.
Lynn,
Yeah.
I've put my finger in the ultrasound beam and not felt anything. I'm just wondering, what did you have to do in order for it to be felt by the subject? Or did they did they have a perceived feeling?
Yeah, so I think, so the receptors in your fingertips have tuning curves. So you have to pulse it at the optimal frequency of your mechanoreceptors. So in this case, I think the tuning curve for these, the Meissner corpuscles in the finger is somewhere around 50 Hertz or so. So if you pulse it at 50 Hertz, then you'll get these responses.
I see. And so what I was doing was with a diagnostic ultrasound scanner. And so there's a really short pulse, and it's not pulsed at any particular frequency. It's just
And I'm sure, yeah, because ultrasound itself is we're not, our mechanoreceptors are not tuned to those frequencies. They're way too high.
Mm-hmm.
So you have to pulse it.
Okay. And what was the total duration of the 50 Hertz pulsing?
Yeah, good question. What did we use? I'll have to look it up. I don't recall offhand. It would have been under a second.
Okay.
And then the other thing though, and the pressures were really high too, to do this. And I'll have to look up the paper to see what they actually were.
Okay.
Okay, so let's move back. So that was kind of some background on using somatosensory evoked potentials as a proxy of brain response. And so what we're showing here is we're back to the use of ultrasound for transcranial stimulation. And so what we're showing here are some models of the size of the ultrasound beam overlaid on the cortex. So this is the primary somatosensory cortex, S1. This is the postcentral gyrus, primary motor cortex, M1, and then the central sulcus here, CS. And you can see the size of our beam you can measure it with the size of a gyrus. And then, so here's the model in a coronal view and then blown up down here. And so I've overlaid where these potentials are generated, if you recall from the previous slide. So the N20 would be down here in the base of the central sulcus, area 3A, 3B, the P27 and the P50. And here's our overlaid beam, and you can see that it's highly specific to this posterior bank where these potentials are generated. And so let's deliver the ultrasound there, and it should, well, affect these potentials. So this is our data here. It's somatosensory evoked potential here on the right. So on the y-axis is microvolts, and on the x-axis is time in milliseconds. And then the timing of the ultrasound here is the cyan bar. So we started at 100 milliseconds prior to the stimulation at the wrist, just so that we had under the assumption that the ultrasound energy was always going into the brain, and then you probe it with the median nerve stimulation. So, and then you compare the condition of while the brain's getting ultrasound versus when it's not getting ultrasound, what happens to these potentials. And so the average SEP under the ultrasound conditions in red, and then the sham condition is in blue. And then what we found was the gray bars represent where we saw statistically significant differences. And so we did get an attenuation of this P27 potential here, shown here, which is generated here. And then there does look to be an effect. I haven't put the N33 up here, but the N33 would probably be generated somewhere in here. And so we saw an effect there, and then not so much down the processing chain. And the other thing that we can look at with EEG data is to look at kind of the spectral power of the data. And so EEG is traditionally characterized by different frequencies that make up kind of the biological signal. And these can be in the alpha, beta, and gamma range up to about 55 Hertz. And so we can just look at the time domain now in the frequency domain. And so what we show is that there's high power in the gamma and some alpha and beta under the normal conditions, but in the ultrasound condition, it attenuates the power of these frequencies. And what I'm showing over here in gray and maroon is just the statistical difference maps between ultrasound and sham, which are eyeball apparent between these two figures here.
And what is the meaning of the attenuation of those frequencies?
So this would, whatever the generators are that contribute to the power in that signal for these bands, have been attenuated or muted. And so the one thing that I didn't mention, I'm actually glad you brought it up, was that it's not 100% clear if a somatosensory evoked potential represents the serial processing chain or is actually just averaged oscillations in these different frequencies. And so if you saw an attenuation in any one of these kind of peaks, you'll always see an attenuation in a power. And so if I look at the power of this signal, the frequency of this peak here has some kind of power. And so that attenuation is reflected in kind of the attenuation of that power. But these are evoked powers. So I don't think there would ever be a case where you'd see an increase in the potential size and a decrease in the power. They kind of go hand in hand.
So I have a similar question. When has anyone characterized the operating frequencies of the different cell types in the cortex, or do they all fire sort of synchronously if they're local to each other?
Yeah, so if you have just a random ongoing EEG, that's difficult to do. When you send in a electrical pulse at the nerve, and now you get this evoked response, you can look at the contributions of these areas to these frequencies. But I don't know offhand exactly what populations contribute to which frequencies, if that's what you're asking.
Mm-hmm.
Then the other issue with EEG that we'll get into a little bit later is the I mentioned how you can source localize these EPs, but in general, EEG does not have very good spatial resolution. Certainly if you're looking at just ongoing or not time lock EEG, it's very difficult to spatially localize, just due to kind of some inverse issues in getting that, recording the signal from the scalp through the skull. The skull is just basically a filter and it blurs it. So it's very difficult to do these things, which is why we did evoke potentials and not just kind of general EEG. But I think there is some research out there, and we've showed that the ultrasound effects do look to be preferential to gamma effects versus slower frequency effects, but the reasons for that aren't clear yet. So the next thing that we asked is, because the ultrasound, the lateral resolution, spatial resolution of ultrasound is so fine, that we wanted to test that spatial specificity in this experiment here. So what we did was we translated the beam from the original position in primary somatosensory cortex, either 1 centimeter anterior or 1 centimeter posterior, under the hypothesis that we are no longer aiming where those potentials are generated. And so we should not see an effect on those potentials anymore. And so that's what we're showing up here. We did not see any statistically significant differences in the two traces here between sham and FUS. And then if you look at the power images down here on the right, there were some small statistically significant differences later in the trace, but you can see even just visually between the two traces as compared to the last frame that we didn't see significant differences. So this is some evidence attesting to the spatial specificity of ultrasound. So we found a physiological effect. And so the next question was, well, does this translate to behavior? And so because we were studying the somatosensory cortex, we decided to do some tactile discrimination tests on the fingertip. So I showed you in a few slides earlier that the information that goes into 3A and 3B, where we aim the ultrasound, a lot of information comes from the fingertip. And so we tested some behavioral tasks on the fingertip. And so on the top is a 2-point discrimination task, and on the bottom is a frequency discrimination task. And so in this task, random pin distances are pressed into your finger, and you would have to report whether you felt 1 or 2 pins. And so what we're showing down here on the x-axis are these different pin differences. And we all have a threshold for where we'd say we'd feel 1 or 2 pins. And so that's what's represented here by this horizontal line. And so in most people in the sham condition, so their threshold is roughly around 2 millimeters. So if the pins are spaced closer than 2 millimeters, they would say 1 pin. If it's more than 2 millimeters, they would say, well, I feel 2 pins. And so we did this under an ultrasound condition and under sham condition. And what happened was the people's threshold got lower under the ultrasound condition. So they actually got better at the task. So they could discern a pin difference of roughly 1.5 millimeters under the ultrasound condition as compared to 2 millimeters under the sham condition. So we thought that was quite remarkable and didn't 100% believe it. So we did another task. And so this was an air puff discrimination task down here in the bottom. So I delivered 2 air puffs in sequence, and they have different frequencies, and then you have to tell me whether they feel the same or whether they feel different. And then you change the frequency between the air puffs down here by some kind of frequency. And so these are frequencies that our fingertips are highly tuned to. And then once again, we have the ability to discern frequency differences to a certain extent. And so what happened here is in the sham condition, most people could not discern a 50 Hertz difference. But under the ultrasound condition, they could discern a 30 Hertz difference. So this again showed that it improved their tactile acuity. So this was a very interesting finding, and there was a lot of debate internally in the lab and also externally about, well, how come if the somatosensory potentials go down, their behavior gets better? And we don't have a good answer for that, but I don't think anyone has a good answer for that because it's never really been established that a decrease in an evoked potential is related to, say, an attenuation of brain response or ability. So there's no real relationship between EP amplitude and behavior. So an interesting finding that's definitely worthy of discussion.
Wynn?
Yeah.
In plot B, if we're looking at the sham condition in blue, so in order to be able to distinguish the frequencies as different, they have to be lower frequency. Is below the line, below the 1, being able to distinguish them as different?
Above the 1 would be
different.
Being able to distinguish.
Okay, and then what do you have to do in order to be able to distinguish? It has to be higher frequency than 50 Hertz?
The difference between the two stimuli has to be for the blue would have to be more than 50 Hertz.
I see.
So the variance around this 50 Hertz. So in the sham condition, most people can determine a 50 Hertz difference on their fingertip, but could not determine less than that. It's too fine a difference.
Okay.
But in the ultrasound condition, they did fairly well in terms at all frequencies that we tested.
Okay.
Does that make sense?
Yeah.
Okay. So that was our first study using EEG. The other thing we were interested in was to pair ultrasound with MRI imaging. And we think this is a nice combination because you can take advantage of MRI's whole brain, high spatial resolution. And so I think the technologies are complementary. And in this case, we're using bold imaging. Did you guys discuss any of this in this
We haven't
course?
really talked about bold imaging yet. No.
Yeah, so I thought I'd put in a couple quick slides, just very basic about what does a bold image represent, and so why do we choose to look at this? And so the idea is that there's this neurovascular coupling in the brain, and so neurons need energy, and the energy is supplied as oxygen through the blood. And so we shouldn't get too tied up in the old and the new here of what leads to the bold response, but there's neural activity and there's energy demand, and then you can measure that through this energy supplied through blood volume. And so we measure this bold response, which is this blood oxygen level dependent response, which is a ratio of the amount of deoxygenated hemoglobin and oxygenated hemoglobin in a specific volume. And then, of course, so neural activity will alter this ratio, and then this is what you measure with bold. And so if there's neural activity, you get an increase in blood flow because you need oxygen for energy, and then that brings in an excess of O2, and then so the deoxyhemoglobin levels fall, and then that's what the MR signal really is representing. So we wanted to see if you deliver ultrasound, does it affect this kind of neurovascular coupling? And one of the reasons we were interested in this is because this has been shown in animals. And so I think you guys have already seen the lecture of Sun-Shik Yu. This is his rabbit study up in the top. So they did this in craniotomized rabbits. And so this is the hemodynamic signal change on the y-axis. It's always usually expressed as a percentage of baseline. And then this is time. And so this is a very slow response over seconds. And so what they showed, so if you just randomly sample a volume, this is what you get in green. There's no real change. But when they delivered ultrasound at 2 different intensities, they get this increase in this hemodynamic response change. And 1.5%, I mean, it seems like nothing, but that's a very standard change. It's a quite robust change. And so they showed that they got this change where they were aiming. So their blue crosshairs where they were aiming the ultrasound. And I think, Keith, you brought up in that lecture, because he showed, and I thought it was a great question. Remember, he showed bilateral paw movement. And then Keith, you asked, well, how come he didn't get bold on the other side as well? And so, well, I don't know, but I think it's interesting that they got bold on one side, but they got the bilateral effect. Anyways, that conversation for another time. So down here on the right is some work done by Charles Caskey at Vanderbilt. That they did this in primates. And I think what's important to show here is this the hemodynamic response function here over time. So anytime there's a pink bar here is when the ultrasound is on, and then the white bar is when the ultrasound is off. And so you can see that this response function very nicely follows this on-off trend. And so they're showing here ultrasound is in blue, and then a tactile stimuli we kind of talked about. If I just poke you on the fingertip, you're going to get that brain response as well. And so their ultrasound response is very similar to if I just tapped you on the fingertip. So I think this was a quite convincing data plot in my opinion. And what they're showing on the bottom is if you don't do anything, you just kind of get this random signal. And so Unfortunately, so we've been doing this. I did a lot of this work at Virginia Tech with Jamie Tyler that we wanted to do this in the scanner. And we have some instances where we can get a fairly localized bulge response, but most subjects, we didn't get any response. So we published these results in 2016, but we have lots of data where it's just, for us, it was just not a robust response. And what I'm showing down here in this rendering of the subject in the bottom is that in this case, this subject had both increases and decreases in the bulge response to the same stimuli, which is very curious. And you can see in the rendering, you can see the outline of the transducer where it was placed. So in this case, we were directing it at the primary motor cortex. So we were not able to, in my opinion, with any kind of robustness, reproduce those animal results.
Did you do that in combination with a stimulation, or was the ultrasound by itself?
Yeah, in that case, it was by itself. So you're just trying to use the ultrasound itself to elicit a brain response. And we had difficulty doing that. So what I'm showing here in this slide is we induced a response a different way and see if ultrasound modulates it. So it's a very similar strategy to what we did with the EEG and the evoked potentials, but now we're using fMRI. So in this case, we had, if I have you tap your finger or move your finger, you'll get a response in the primary motor cortex. And so that's what we're showing here. Is if I ask you to move your, in this case, it was your thumb. If you move your thumb, I can record a very localized response in the brain, in the primary motor cortex. And so we want to see if we direct ultrasound at that finger or thumb representation, can we up or down regulate it? And so what I'm showing here is just this is an empirical measurement of the beam that we did in our acoustic tank, and now I've just overlaid it to scale on the brain. So you can see that ultrasound has a resolution of an individual finger representation, and so that's a real nice advantage of ultrasound. So here I am doing some pilot work in the study. This is our basic setup. So we had subjects either move their thumb, middle, or index finger in these 30-second blocks. And then we delivered ultrasound at varying intervals. So it was kind of an on-off kind of design. And this was these are our ultrasound parameters. I believe you guys have talked about these in prior lectures, so I won't get into it in too much depth. But these parameters are the same parameters that we use for the EEG studies. So here's from one subject, this is their thumb activity when they move their thumb. Here's the activity when they move their index finger, and here's the activity when they move their middle finger. And I just overlaid our ultrasound beam to show that we can get these resolutions with ultrasound. And so what we did was I've displaced the ultrasound here just to show the relative sizes. But for the experiment, we put the ultrasound over the thumb representation or either one of these representations. So if you direct ultrasound at the thumb representation, it should affect that one and not the other two. And then the same for the other fingers. But once again, we didn't see a change in that hemodynamic response function. We didn't see a change in the magnitude at all. The only thing that we found was in this case, was an increase in volume of the size of the representation, which is shown here in 5 subjects. So these are all the sham conditions here, and under the ultrasound condition, in all 5 subjects, that response from a finger movement expanded. So once again, we're having trouble. The interpretation of this is difficult, and the reasons why we're not getting magnitude changes in fMRI is not 100% clear.
Wynn, is it possible? So I like the positive control of having them actually tap the finger, but would you expect to also see your EEG or your transcranial electrical recordings to show a response under these conditions?
Say that again.
So earlier you showed that there was a lesser deflection in some of your signal
Right.
from your EEGs. If you had hooked those up during this experiment, would you expect to see that reduced signal as well?
Yes.
As
So if
sort of
we did
another
simultaneous EEG, which I think is what you're asking, yes, I think
Mhm.
because the parameters are the same.
So
But
do you think there's like a threshold at which Bold can detect changes and that your changes are below that, or there's some fundamentally strange
yeah,
thing going on?
so our delivery, yeah, I think these are all possibilities. So even though our parameters are identical, our delivery strategy was different because there's some limitations when you're in the MR when you can deliver it. And so in this case, it was interleaved with acquisition. So it could be that we just did less total ultrasound. You're also right that it could be that the Bold signal is a little less sensitive to pick up these changes. These are all possibilities that why we're not picking it up. But I struggle to find, like, I don't have a definitive answer as to why. It might also be that, yeah, the Bold signal itself is not optimal to pick up these small changes. I'm not 100% sure. I wish I had a better answer for you. Okay, we're going back to EEG here. And so I mentioned earlier on in the lecture that one of the benefits of ultrasound versus other non-invasive technologies is that you can deliver it deep to the brain. And so we wanted to test this out in humans. So it had been shown in pigs, done by Jeff Elias's group here at UVA prior to when I got here. They showed some very nice data that they could stimulate individual nuclei in the thalamus of the pig. And so we wanted to test this out. That was possible in humans with a single element. And so we took the same strategy as we did when we were investigating the primary somatosensory cortex. Because there's this nucleus in the thalamus here in green called the ventral posterior lateral nucleus. And this is this nucleus here in this dorsal column medial meniscal chain that you can record through scalp EEG as this P14. So this is a very nice opportunity that we can record something deep in the brain non-invasively through scalp EEG. And so our theory was, let's direct ultrasound where this potential P14 is generated here in the thalamus in green and see if we can specifically modulate that potential. So we had a very similar setup to when we did this in the cortex. We continuously recorded EEG, in this case from 6 electrodes on one side of the head, and then delivered ultrasound prior to the median nerve stimulation. Not 100% sure if this is true here, that we delivered it 2 seconds before. I don't know, this is not right. Ignore B over here. This is what we did over here. Sorry about that. It's the exact same strategy. We delivered 500 milliseconds of ultrasound that started before the median nerve stimuli and lasted up to 400 milliseconds. And in this case, same parameters as we did before, but a slightly different, we had more total stimulations, and in this case, we had a shorter ISI. So you would deliver 500 milliseconds of ultrasound, you wait 4 seconds, and then you deliver it again. And in this case, we did 300 stimulations of this for each participant. And then, so to localize this nucleus in each subject, you can do a, in this case, this is a diffusion weighted image from an atlas that we took in the publication, and then you can morph that onto the individual subject MRI, so you get a good idea where their VPL is. And then we use that as the target. And then we designed a transducer that would overlap with that target. And so we decided to target the thalamus through the side of the head. I mean, technically you could go from anywhere in the head, but this was the side of the head is advantageous because the bones are thinner and flatter. And then this was the shortest distance to the thalamus on the head. And so here's the average numbers for all our subjects. We had 40 subjects. And so our target was on average 57 millimeters from the scalp surface to the target. And so then we developed a transducer that had its field at that depth. So the face of the transducer is here, and then here's the field here. Here's the lateral extent of the field, and then here's some line plots of X, Y, and Z, the size of the field. This was all data that we collected in our acoustic tank. And then we ported this data into our acoustic model, showing how, in this case, in one subject, that going through the temporal window, we do maintain to a very decent extent the lateral resolution and also the same kind of beam geometry, even when you take into consideration skull effects. So once again, we use somatosensory evoked potentials. And so this is the data from the 5 channels recorded at the scalp, and the ultrasound condition is in red, and the sham condition is in blue. And so in all 5 channels we recorded, in this case, so here's the P14 potential here. In all 5 cases, the ultrasound condition attenuated, made this potential smaller compared to the sham condition. It's not too surprising that all 5 channels show the same thing because of the EEG. If channels in close proximity will generally show similar things. But what I think is also interesting from this data is, unlike the cortex data, where we generally didn't see an effect on these later potentials, we did see an effect on these later potentials in this case. So it's possible that if you disrupt the activity of the information in the thalamus before it gets to the cortex, it might change the cortical response as well as the thalamic response. So we thought that was kind of cool. And once again, we also saw a change in behavior. In this case, we just did that 2-point discrimination task. And in this case, we did not see a difference, a behavioral difference between FUS and sham. This bar graph over here on the right looks like there's a difference, but this is only really a difference between FUS and chance. So we didn't see a statistically significant difference between FUS and sham. So our physiological results in this case, did not translate to behavior. Okay, so now we're going to talk about delivery of ultrasound to the primary motor cortex. So I think in previous lectures that the students have seen evidence, or I guess from evidence, that if you deliver ultrasound to the primary motor cortex in a small animal model, you'll get a tail or a paw twitch. Is that right? Are they aware of this?
Yes.
Okay. So that doesn't seem to happen in humans yet. I'll say yet because well, I don't know why, but we can talk about that if you'd like. But so if I deliver ultrasound to the primary motor cortex in humans, I don't get this finger response or an arm response, or my tail doesn't move. So I need, if I'm going to test the effect of ultrasound to the motor cortex, what do I measure? You could do behavior, but you'd like to have a kind of a more discrete physiological or biomarker. So in this case, what we decided to do is we decided to use TMS to do this. So do you think your students are familiar with TMS? Should I show this video?
Yeah, go ahead.
Okay, so this is just a brief video done by another lab, but I thought it was so great that I didn't have to do it myself. So let's see if this translates.
Hi Recon, welcome to our laboratory. Today we're going to show you how the transcranial magnetic stimulator works. So first thing, Yoshi's going to put the magnetic coil over top of the area of my motor cortex that's responsible for the muscles of my hand and forearm. Once she's there, she's going to deliver a magnetic pulse. Oh, and you probably saw my arm twitch there. And we can also see this response over here on the oscilloscope because we've measured it using the EMG electrodes over the muscles of my hand and forearm. Now what we're going to do is move that magnetic coil over the span of my motor cortex from one side all the way over to the next. And what you'll see is muscles slowly recruited from my hand up to my elbow, shoulder, right leg. And after she switches hemispheres, we'll see some of the left leg come in and then down the muscles of my left arm. So here we go. So right hand, little bit of elbow, some shoulder and leg, still right leg, little response in my left leg there, we've switched hemispheres, more leg on the left side, left shoulder, left elbow, and left hand. Thanks for joining us.
Love that video. I think it was extremely well done. So this is one of the benefits of TMS, is that there's an overt biomarker that you can record. If I put an EMG surface electrode over these muscles and deliver TMS to the area of the brain that represents that muscle, I can record this motor evoked potential, which is shown here. And so under different manipulations, just like we talked about in evoked potentials, that you can increase or decrease the amplitude of this thing as a proxy of increase or decreasing the excitability or activation of the cortex. And so this is a study that we did in 2013, which is not ultrasound related. But if you we made a grid on the head, and then you deliver TMS to each point on this grid. It's spaced by 1 centimeter. So this is a 5 by 5 centimeter grid. You get, and we color coded the amplitude of the MEP response. There's always a little hot spot of where that muscle is represented, and then it has some kind of, distribution around it. And so you can see that. So TMS does not have really centimeter resolution. I can stimulate this muscle out to several centimeters around it. But I mean, it's better in one spot than the other, but you still get it because the energy distribution, as I showed in our model in a previous slide, covers quite a larger area. So we wanted to see what happens to the MEP that we generate with TMS if we deliver ultrasound to that area of the cortex. Whoops. So we came up with our cool ultrasound TMS design here. So what we did was this is the TMS coil here in beige. They call these a figure 8 coil. I won't get into why, the physics of this. But then we 3D printed a little holder and made our custom-made transducer that we stick right in the middle of the TMS coil. And then this is our stereotaxic tracking because we use MRI neuronavigated stereotax so that we can position this on the person's head exactly where we want it to be. My labels here are off center, that's no big deal. So then we did some models of, here's the electric field generated in the head from TMS. And you can see that we can target the primary motor cortex fairly well. It changes when with our design, which is not too important for today's lecture, but the point is that we still get the motor cortex, but don't be too worried. I shouldn't even have put this in that as a talking point, because then you're like, well, it changes with the ultrasound. The spread of the field changes with the ultrasound, but the field itself, the response in the head doesn't. I mean, and this is not, ultrasound hasn't been turned on yet. This is just, if you take the coil, if you offset the TMS coil from the head, because it's got a depth focality trade-off, now the field, because the coil is removed, the field is spread out a little bit more. But the point is that you still get the motor cortex. Here's our acoustic model of the ultrasound. And so this is the central sulcus here in the white line. And so here's the size of our ultrasound beam directed right at the muscle representation that we're interested in. In this case, it's just a muscle in between the thumb and the index finger called the FDI, first dorsal interosseous muscle. And the reason you choose this muscle is because it's easy to find and you get a large, robust response. So what we did was one of the strategies is you can do these recruitment curves. So if I put the TMS coil on your head and I started at 50% intensity and turned it up to 100%, the more energy I put in your head with TMS, the bigger the muscle response. And so that's shown here in this curve here in black. And so this is the MEP amplitude in microvolts on Y, and then this is our TMS output on X. So we did it with ultrasound and without ultrasound. And so the reason that we paired ultrasound with the coil is we wanted to do it concurrently in time, because a lot of our other results always showed that the effects always seemed to happen during when ultrasound was on. We could have done a pre-post design, but we thought that this, we like this idea better. And I think there has been one publication since ours exploring the motor cortex, and they did a pre-post design. But in this case, we're delivering it concurrently and concentrically. And so our general finding here was that under the ultrasound condition, the amplitude of these MEPs was smaller as compared to no ultrasound. And you can see this here. This is some data from one subject. So you can see at a TMS percentage of 50%, you get a small MEP, and then it gets bigger. But under the ultrasound condition, it's generally across the board smaller. So suggesting that in this case, similar to our somatosensory cortex, that the effect here was inhibitory. The other cool thing about TMS is that you can manipulate TMS's parameters to do different things and to affect different motor microcircuitry just with TMS alone. So in this case, if you deliver 2 TMS pulses at some temporal spacing, you can either induce inhibition or you can induce facilitation. And so there's a lot of literature on, by doing this, what are you affecting in the motor cortex, and whether these are kind of GABAergic type or glutamatergic type or NMDA type of populations of cells. And so it gave us an opportunity to see what does ultrasound do to either natural inhibition or natural facilitation from TMS. And so that's what, again, what we're showing here. So if you deliver TMS at short intervals, it's inhibitory. Longer intervals, it's excitatory. And so what does ultrasound do to this? It did not result in any further inhibition, but what it did do is it inhibited the facilitation. And so this data up here in this line graph is represented down here in this bar graph. So this is the short interval cortical inhibition, and this is this intracortical facilitation. So we dampened the effect of the facilitation. But what should be noted is that it didn't totally remove it. So there's still facilitation. We're still above baseline level here, normalized value number 1. It just kind of dampened it. Okay, so how are we doing on time, by the way? What time?
I don't really
know. I don't know what time we started.
Yeah, I don't really know what time we started either, but it's probably getting towards the hour.
Okay.
But on the other hand, this is really great. So yeah.
You can edit out all the bad parts, I suppose, afterwards.
Okay.
So I think if ultrasound is to translate to a clinical therapeutic, I think, and we can debate this if you'd like, that the effect should likely outlast the stimulation. And so this is how TMS is FDA approved as a depression therapy. And what they do is they deliver a lot of TMS over multiple sessions, and it has some kind of lasting effect. And so I imagine that if ultrasound is also to translate to a potential effect, that you would like the effect in the brain to outlast the stimulation. And all the data I've shown you up until now is that the effect is always while the ultrasound is on. And so the pairing of ultrasound with TMS is actually kind of nice, that we can now probe the lasting effects of ultrasound actually quite easily. So you deliver ultrasound at the same time as TMS, and you get this inhibition. This is represented by this bar. But now, if after I turn off the ultrasound, I can probe the motor cortex at different latencies afterwards, and that's what I'm showing here. So 100 milliseconds out to 10 milliseconds after the offset of ultrasound. And what happens to the amplitude of the MEP? So while the ultrasound is on, it's attenuated, like I showed before. And then this attenuation starts to go away. In this case, it looks like it kind of lasts about 500 milliseconds, then it just kind of, it goes back to baseline. So the question is, how much ultrasound do I have to deliver and how for the effect to protract? So in this case, we delivered 500 milliseconds of ultrasound. Here's 1,000, here's 1 second of ultrasound, 5 seconds of ultrasound, and now 5 minutes of ultrasound. So I went through those pretty quickly, but we can go back and look at them. So there's 500 milliseconds. In this case, the only statistically significant change was actually while it was on. In this case, this is preliminary data, so you can take it with a grain of salt. Here's the baseline normalized value, and it looks in this case that we're getting about 2 seconds longevity out of a 1 second pulse. A 5 second, this is not a single pulse. This is not continuous ultrasound. This is pulsed at our pulse repetition frequency. It looks like this seems to last 5 seconds of ultrasound. In this case, seems to last for about 10 seconds. And then 5 minutes of ultrasound, we've measured out to 10 minutes, and at least looks to have a lasting effect. So it looks like if you increase the duration of ultrasound, we didn't change the intensity, or we didn't change any of the other parameters, we just changed the duration as a starting. We need to start somewhere. You could probably, I think our next attempts will be for intensity as well. So it looks like these results kind of mimic the TMS literature, that if you deliver TMS for longer, the effects seem to last longer. And maybe this is just a basic principle of neuromodulatory effect. Of more energy has a longer lasting effect. So in the motor cortex, we did a simple reaction time task as well, because it's always nice if you can get a physiological change to also get a behavioral change. This was a very simple Q reaction time task. And we did 2 different shams. So here's ultrasound here. We did a sham over M1, and we also did an active sham. So we actually delivered ultrasound to another area of the brain that is not supposed to be involved in the task. So in this case, they had to react with the muscle that we were targeting. And so what we found was a very modest decrease in reaction time under the ultrasound condition compared to the 2 shams. So once again, this is an inhibitory physiological effect, but it has some kind of it's behaviorally they make them behaviorally better, which is curious. And so other groups have also shown similar effects, and certainly some of the work in the primate, when they were looking at eye latencies. I'm not sure if you've gone over that data yet, but they always see a decrease in latencies, an increase in behavior as well. So do we have a little bit more time to discuss? This is some of our current work.
Yeah.
Okay. So we've been doing a lot of work non-invasively, but we have an opportunity that I'm working with a neurosurgeon at UVA that we're really interested in what the cellular effects are in humans from ultrasound. And so a lot of the animal work and the cellular work is very valuable, looking at how ultrasound affects things on the channel or molecular level or the cellular level. But I think you've probably discussed the effects, some of the confounds of small animal models. Certainly anesthesia is an issue. Have you discussed cranial vault sizes and some of these issues relative to the beam? Probably.
Well, we talked about sound bouncing around within the cranium, sort of generally, but not specifically.
Right. So we thought that we have an opportunity, if we could remove some of these confounds by directly testing this in humans, that it would be nice that we wouldn't have to deal with any of these potential confounds like anesthesia or potential scaling confounds between small animal models and humans. So in this case, these are patients that are undergoing DBS surgery for movement disorders such as essential tremor or Parkinson's disease. And so they will have implanted electrodes in the brain. And so we can measure the cellular response, individual cells, and also local field potentials under conditions of, in this case, ultrasound. And so we've integrated the transducer into the stereotaxic neurosurgical frame here, where we can look at these effects. So I'm showing you some data here from this is the subthalamic nucleus. So this is one of the areas that they would, in DBS surgery, stimulate to remove some of these movement disorder symptoms, which is usually to alleviate tremor symptoms. And so you can see here's some images showing the implanted depth electrodes. And then what we do is, again, we go through the side of the head here to target ultrasound to where they're recording. And so here's one recording from one subject. And so you can see that we're pulsing ultrasound here. This is our ultrasound artifact. Here's individual spikes. And in this case, so LIFU, of course, I've used another acronym again, low intensity focused ultrasound. But the point we're trying to show here is that we can record these spikes in between these pulses of ultrasound. And so what does ultrasound do to the spiking activity? So here's data from one subject. So these are all the spikes we recorded. We have about 30 seconds here of data, and then the red line represents while the ultrasound was on. And then we have about 3 minutes of data after the ultrasound. So in this case, similar parameters to what we did non-invasively, but the big differences are that this is the 5-minute protocol that we use. So this is 5 minutes of ultrasound. This is so 500 milliseconds of ultrasound every 3 seconds. And what we found in this subject was we found an increase in the spiking. And you can see here, the ultrasound turns on, the spiking frequency goes up, and then you turn the ultrasound off and the spiking frequency goes down to the original values. So I thought this was quite remarkable data that we do see this time-locked single cell response to ultrasound.
So Wynn, is, sorry, for the roster plot, is each line here an individual trial of one patient or many patients? And are they continuous?
This is the data over time. This is this spiking data over time. And then just the spike, interspike differences were just converted to Hertz. Does that make sense?
Yeah, I think so. Well, oh, I see what you're saying. Okay, okay. I might come back to this with more questions.
Okay, please do. So the other thing we can look at is the local field potential activity. So here's our raw data up here. You can see the ultrasound artifact. And then if we do a simple wavelet convolution, you can see the strong power in 20 Hertz. And this is quite common in these patients, that the activity of the STN has a strong kind of a power in this beta range. And then while we turn the ultrasound on, we saw a shifting of this power into a roughly 30 Hertz. And that once we turn the ultrasound off, in this case, it stayed elevated. And so we've just blown up the pre and the post here, showing the power. And then we just did, here's an FFT of this power over time, showing that it did change from 20 Hertz at baseline to 30 Hertz after ultrasound. Unfortunately, there's not a lot of time in the operating room to continue it to see how long this lasts. But in this case, the power didn't return to baseline, but we did see a change in this power as a result while the ultrasound was on. The other thing that we were able to do here at UVA in the neurosurgery department is we also have epileptic patients that have cortical implants for electrodes. And so we can look at what the effect of ultrasound is in these local field potentials. So this is data from one epileptic patient who's undergoing invasive epileptic monitoring. And so we have an opportunity to test out the effect of ultrasound on these implanted, on this activity from these implanted electrodes. So in this case, we delivered the same protocol. This is 5 minutes of ultrasound at this ISI of 3 seconds. And so here's the data here. Here's 5 minutes of baseline data recorded from a cortical electrode that we're able to target using MRI navigated ultrasound. Here's the data under the fuss condition, and then here's the data after. So each one of these is 5 minutes. And then we're looking at the frequency range from, it's probably 2 Hertz to 60 Hertz. And so this data up here at the top is all this data collapsed over time. And so the average power before ultrasound is in black, ultrasound is in red, and then after ultrasound is in blue. And you can see we have a general decrease in the power, in this case, in the frequencies below 10 Hertz. And these are the frequencies that dominate the power spectra for biological signals. But if we can use wavelets to look at these changes over time and what happens. Does ultrasound work right away? Does it preferentially affect some frequencies and not? And what you can see over time is that you get this, it didn't happen right away, but we eventually got this attenuation here that I've highlighted in this red box, that we get a decrease in these powers, and this is what's likely leading to this change here. And then after the end of ultrasound, it popped right back up. And I think that this data here in the next slide is a different interpretation of this previous data. So we've segmented the frequencies into what are traditionally considered like the accepted biological frequencies. So theta, alpha, beta, and gamma frequencies. And so here we have power in dB here on the y-axis, and here's our time. This is the 5 minutes of time here. And then what I've done is I've just binned the powers for the 3 conditions. So pre in black, ultrasound in red. I apologize for the thin lines. And then after ultrasound in blue. And you can see in the red lines that there's this kind of slow attenuation that seems to be most pronounced in the alpha frequencies, but also in beta and maybe not so much in gamma. Then you can look at the histograms of this data as well, and you can see the shifting to the left of the power in these frequencies. So LIFU here is in this kind of peach color. So no real changes here in gamma, maybe some slight shifting in beta, but more, it seems that in this case, ultrasound seems to be affecting kind of alpha and theta band more than the other frequency bands. Okay, almost done. So one of the things I'm interested in is because I come from a rehab background, or partially rehab background, if I see a physiological change, I'd like to see a behavior change as well, because then it has clinical importance, I suppose. And so one of the things that's done with ultrasound, I don't know if you've talked about this or not, Kim, is high intensity focused ultrasound for ablation, I guess. Mm-hmm. And so this is an FDA approved technique to treat essential tremor. And so this is the use of ultrasound to create thermal lesions. And I won't show you the video, but this is an effective treatment to remove tremor. Do you think your students would be interested in seeing the videos?
Well, we have seen this one,
Okay.
Not those specific ones, but we've shown other videos. So
basically, we wanted to see if you deliver low intensity focused ultrasound, does it have any behavioral effect in these patients? And so we have access to these patients before they go in for the HIFU ablative surgery. We have an opportunity to test some low-FU protocols or some LIFU protocols on these patients to see if there's any effect on their tremor. And so what we do is, again, we're delivering this 5-minute protocol, and then we're doing accelerometry. So we have a gyroscope and accelerometer on the hand, and then we can monitor their tremor before and after a 5-minute low intensity treatment. And so this is the data I'm showing here. So before ultrasound is their tremor in black. So if I put an accelerometer on their hand and they put their hands out straight, they have a tremor. And so that's what, here's their tremor frequency here. So this is the raw data. This is 7 seconds of raw data. And you can see the tremor frequency in an essential tremor patient is roughly around 5 Hertz. So this is oscillating at 5 Hertz. And so that's shown here in the frequency plot. And then we will deliver 5 minutes of ultrasound. And then we'll record the tremor again right afterwards. And then in this case, we also recorded the tremor out to 10 minutes. And in one patient, you can see that the tremor power really went away. So you can see it in the raw data here, in the amplitude, and also in the power spectra. And then at 10 minutes, it's returning. So this is data from one subject that was pretty encouraging. So this is the power data shown here in a bar graph. And so we just took the average over 3 to 6 Hertz. So if you just take this window over the main tremor power, you can see an accelerometer. This is in the 3 planes of accelerometry, X, Y, and Z. And then the different colors are before ultrasound treatment in black, immediately after ultrasound treatment in red, and then 10 minutes after the low intensity treatment in blue. And then this is a standard pen and paper test that these subjects would take before and after the HIFU ablation. And so you can see this, and they do the Archimedes spiral. You can see the tremor, and then they'll do a line drawing as well. So this is a line drawing before our low intensity treatment, and this is it afterwards. And I would argue that this doesn't look much better, but this one was considerably better, and also slight improvements here in the line drawing as well. So that's pretty exciting, that even low intensity can have some brief transient behavioral effect in a clinical population. So this is an active area of study for us. I think we've collected 15 patients or so. Admittedly, not every patient looks like this. We have non-responders as well, and this is unfortunately quite common in the non-invasive brain stimulation world, but encouraging preliminary data.
And that would be largely temperature independent. I'm trying to sort of guess what temperature you would expect from this.
Yeah, so if we model this waveform, we do not, it does not get over 1 degree centigrade temperature change. And so this is the estimated in the head. So we're delivering, I think it's 16 to 20 watts ISPPA outside the head.
Yeah, I'm just thinking of, so Kim has shown this with the X-Ablate device. You know, if you definitely use thermal features to inhibit tissue, Kim, you're raising the temperature, what, like 5, 6 degrees with that device to get the inhibition without tissue?
Yeah.
So this is probably an independent mechanism, which is pretty awesome.
We're guessing, yeah, if you can model these temperature changes, we have no way of measuring these things in vivo, unfortunately.
Well, I mean, you could measure the temperature with an MR thermometry.
Yes, if we did this right in the scanner. We've also tested using the X-Ablate system at low intensity, but it has very limited pulsing opportunities, so we can't pulse it exactly how we want, and these are based on, I think these are just some restrictions of the hardware and the software of just how it's built. So we haven't had a lot of success using the X-Ablate itself. Maybe because the parameters really matter, I don't know. But you're right, in that case, we can measure the temperature, and these low intensity protocols are very low kind of joules. Yeah, we don't see any temperature changes. All right, I made it to the end.
Wow, that was great. I think we've seen so much that was, you know, very different than the other presentations in terms of like being able to see the human work. So, Keith, do you have questions?
Yeah, I had a few, and I always tell everyone, you know, I guess this is a good point if you want to listen or not, you know, I might drone on here. So your data with the electrode implants for the epilepsy patients, I mean, you sort of understated how incredible that power shift was, right?
Oh, did I?
I think so. Yeah. Then if you go to either the next slide or this one, I mean, that's an enormous change in power, right? For a single patient. I wouldn't, I'd be really shocked if you couldn't see this with Bolds. Can you do, I guess you can't do the MRI with these patients because of the metal implants, or?
I never considered it actually. I'm not sure if these electrodes are MR compatible or not.
Yeah, so I keep seeing, and when your work is really incredible, and I want to ask more questions, but I know that one of the struggles is when you're working with human patients is we simply can't get the throughput, we can't change the experimental design after the fact, and we can't cross a lot of these studies over. Can you comment on that? I know that's a really vague statement. Are you just, do you often change your mind after it's over and wish you could redo it, or are you revising as you go? You know, what kind of struggles do you have with human studies?
Yeah, that is an interesting point. So certainly with there's a difference between working with healthy normals versus clinical patients. So in the clinical side of things, we have very limited interaction with the patients. You have to do everything quite quickly. So you storyboard this as best you can beforehand, and yes, you hope for the best. And there's difficult considerations that sometimes you have to decide, well, do I need group statistics? So if it hasn't happened in a couple of patients, do I keep doing it? Do I keep doing the same thing, or do I change it? And sometimes you only have access, maybe we'd have access to a patient once a month, maybe not even. So there's a bit of crossing the fingers here and what to do. And if it doesn't work in the one patient, in the next patient, do we try it again? Do we change it up? Yes, these are issues. It's not so much when you can, if you're doing it in healthy kind of college students who volunteer for the experiments, you'll set this all up beforehand. You know, you'll do your power calculations, say, I need 20 subjects based on what I'm expecting, and you can run through it fairly reasonably. Yes, it's not like studying cell cultures or small animals, but yeah, there's, it's the case where I think you do the experiment and you just see what the results are. And if we've got lots of negative results, I'm not gonna, you know, sugarcoat that. Certainly in our bold data, lots of negative results. I only showed you the one slide there in the patient that had a very good effect on the tremor. We've collected 15 patients, and we've kind of got this 5 it works, 5 it doesn't, and 5 kind of in the middle. And there's actually there's lots of documentation in the non-invasive brain stimulation world of this variability. So I'm not sure if that answered your question, but you—
Absolutely. I think it's worth mentioning, Wynn, and I'm sure you know this, when we do these animal experiments, these things are highly optimized. So we can have years of experiments that just don't work, and you change one, you know, small variable or way that you measure something, and then the effect is highly observable. So I just wanted to point out to the students that when you're doing human studies and you can't get that iterative value, you know, it can be difficult to get where you're trying to go, right? And I think you sort of described that very well.
Cool. Yeah. I as my background is in studying humans, and I think some ultrasound research could really benefit from more human studies. And I think some kind of critical mass is starting to happen, that other labs are starting to do this. And so hopefully they'll find things that contribute to the literature, you know, so not one lab has to do everything. Hopefully we can, you know, you mass up these results to figure out what works best. But yeah, human research can be difficult at times. But I also think the benefit is that direct translatability, that you can find this in a human, then you can find it in a different human, that kind of thing, right? Instead of, well, I got this paw twitch in this mouse, why can't I get this paw twitch in this human? I don't know why. Kim, do you have any opinion on kind of the translation of the results from, say, a small animal to a human?
No, not really. Yeah.
One thing I was noticing, and we've heard this before, it's interesting that all of your studies point towards inhibition, whereas in slice or in some animal models, people will show excitation. And there's always the wonder, well, why is it that inhibition is the thing that always holds up in these human studies? I don't know if you have an opinion or thought on that that's pointed.
Yep, that's a good point. I didn't mention it. It was probably obvious throughout my talk. You're right that we always find inhibition. And when people ask me, like, does ultrasound work? And I said, we're very confident that we can inhibit things. Like, ultrasound seems to work quite well for this. But you're right, I have no evidence, at least in my lab, for excitation.
Mhm.
And I don't know why.
I mean, I think that Sung Shik has talked about being able to, in the sheep model, with his EMG recordings in the sheep, that he's been able to get a response from sonicating motor cortex.
Yeah, I think so. His recent paper that came out, I think he reports under somewhere around 5% fixity rates for excitation, which, I mean, maybe that's what it is. And if that's what it is, that's what it is. I mean, it's lower than it is in small animals. We've tested this in humans, and I can't get 5% in a human. It could be a dosing issue as well. So we're very conservative. We err on the side of safety. We're still in humans, and we have no idea what the minimal level of pressure or intensity is to have a neuromodulatory effect. I mean, we know the max, right? You're always worried about the max from a safety or heating perspective, but we don't know what the min is. And so maybe we're really low. Like, maybe we're right on that threshold for neuromod. We're just getting these effects. Maybe we can't turn the dial.
What's your strategy then? Do you operate right at the FDA limit?
Yeah, our temporal averages, depending on our kind of pulsing scheme, are a little higher than that FDA, but they're lower than the European physiotherapy limits. So yes, we operate in between those two limits. Our PPA is lower than the FDA limits, but our temporal average is higher.
Yeah, so Wynn, your PPA is, way below the limits, which I think are like, it's 170 or something.
Yep.
So one of the discussions surrounding your work and others with the inhibition is that a lot of these human studies just aren't getting, maybe high enough in this instantaneous sort of pressure. Have you considered going above 10 watts per centimeter squared and just keeping the pulse, very short and then non-repeating? Is that of interest or?
Yeah, it's adventurous. We've considered it. We haven't done it yet. I'm not sure. Yeah. I mean, it's on the whiteboard in the lab of things to do, but yeah, we haven't done that yet. I think, I mean, there's a bit of, reticence and apprehension that, we don't want anything to happen, right? Because you don't want to set the field back, or of course, the safety is paramount. So I always reference ultrasound to other neuromod technologies because that was my background as well. And so these are things that certainly TMS struggled with when they were first figuring out how much TMS to deliver. I mean, they induced seizures in people. And that's how people learned how to back off. And I guess that's how we learn, right? This has happened, it's like, don't do it again. I just personally, I guess I'm pretty conservative. I don't want to be the lab that does that, even though that's valuable information to know these things where things might start to get, dangerous. But yes, I think the field I think there's some room to up the intensities. The other thing is that I think maybe ultrasound might work like TDCS. So we didn't talk a lot about this. You can ramp up TDCS as much as you want, and you do not get a motor evoked potential. That technology, just like a broad electric current in the brain, just does not induce an overt excitation. TDCS, how they think it works is just kind of biases membrane potentials it'll bias the system or the circuit to either be more or less excitable. And maybe that's how ultrasound works as well. So TMS evokes an overt motor response because this is a lot of energy in a short amount of time. Whereas ultrasound and TMS maybe just don't work like that. So I used to think when I was training with Jamie that was the holy grail for people to accept ultrasound, is that if I could make your thumb twitch, people would believe it. And that certainly helped TMS gain a lot of recognition when he first did these studies, and it's quite dramatic. If you see the videos and someone's thumb twitches, you're like, that's incredible. Like, yes, TMS works. And Jamie and I would debate, like, is that what we need for ultrasound? That we both agreed that when we were first doing this, that would be very convincing. But we also thought that, I mean, I don't really know that ultrasound works this way. I don't think I can deliver enough energy per unit time to do this.
I mean, well, okay, so I don't want to get too far out here, but theoretically, you know, when you're killing the cell, you're going to get a depolarization at least one last time. And that suggests that, yes, if you go high enough with ultrasound to the point where it's going to kill the cell, you may get a little thumb twitch if that's encoded by a single spike. So again, it's like there's got to be somewhere in between 0 and extremely high that would give you that readout, not that you should ever go near it. And I agree with your philosophy on that. Sorry, can I ask one more question, or Kim, did you want to?
Well, I think we have to turn off the recording at some point here.
Well, the students can, you know, they can stop this. I just had one last.
They're long gone.
All right,
Yeah,
go on.
they might have already tuned off. This is
Yeah,
one of
okay,
the Donatos here.
That's fair.
And sorry, Wynn, you're you know, maybe the world's expert perhaps on combining TMS and focused ultrasound. Do you think about the orientation of the fields relative to each other? Because with TMS, you're drawing these lines and the electrons sort of, you know, come into them, and that may be allowing them to cross the membrane or not. But ultrasound's different because you're pushing perhaps the membrane in another direction. So in theory, you know, doing these things orthogonally, having TMS lines this way and pushing the membrane this way may give you a better response than if you did them parallel, where you're just moving the ions along the membrane, even if you move it. So in your experiments, have you thought about whether or not the fields are orthogonal or parallel? And does that influence what you're doing?
It does.
Or am I just wrong in everything I'm just saying here? It's just something I've thought about.
No. If you deliver ultrasound parallel to a magnetic field, you can induce these magnetoacoustic electric fields. And so, if you wanted to combine the two energy sources to produce a higher electric field in the brain, that's what you would do. And you're right. Right now they're perpendicular, but that wasn't our purpose to summate the fields in this case. To get more kind of, that would de facto be more of an electrical, I think. Well, I suppose it would be, yeah, it would be mechanical, but that wasn't our original intent. And I don't, for ultrasound to have its effect for the field to line up with the magnetic field, I have to think a little bit more about that. But yes, that is certainly a method to induce increased activity. But yes, that's not what we were doing. So we did some safety studies, or just some kind of practical studies prior to applying this to humans, showing that, and we did showing that how the fields may or may not interact to produce either more or less pressure than what we were assuming, or more or less energy in the brain than what we were assuming. And because the fields were perpendicular, this was not a concern. But yes, so they don't interact essentially in this case. But that wasn't our intent, to make a new device, this kind of magneto-acoustic device, but it's certainly possible.
Okay, I think I'm going to turn it off now.
Good idea.